This video lecture is going to start the chapter 7 on diseases and disorders of the blood. It's going to be focusing on diseases of the erythrocytes, the red blood cells. As we head in. The chapter itself is going to break out into 4 pieces. We're looking at erythrocytes, bleeding disorders, leukocytes, diagnostic tests. And it's actually a very straightforward chapter, and so you should be able to get through it pretty easily. Now, blood itself, in general, the average person has approximately 5 liters of blood in their body. Textbooks will argue about, 4 to 6, 3 to 5, 5 to 7, big people, little people, men, women. Just let's make it simple and say that everyone has about 5 liters of blood. That's an easy way to remember. Good landmark. Now, when we look at the fluid portion of blood, we talk about plasma versus serum. Plasma is the liquid portion. We've taken out all the red blood cells, the white blood cells, and the platelets. What we're left is the electrolytes, the proteins, glucoses, vitamins, minerals, so on and so forth. Serum is plasma minus the clotting factor. So if we want to save the fluid for a long time, not take a chance it's going to coagulate up, then we'll precipitate out the clotting factors and we're left with serum. The formed elements, those are the cells and cell fragments. That's the erythrocytes, the leukocytes, and the platelets. Hematocrit refers to the measure of red blood cells in blood itself. So if we take a tube of blood and we spin it down in a centrifuge, we'll find the plasma will sit up on top, and then the red blood cells will sit on the bottom, and in between will be a buffy coat made of white blood cells and platelets. This volume of red blood cells is the hematocrit. And so when we ask for your hematocrit, we want to know what percent of your blood is the oxygen carrying ability. And for the average person, it is about 45%. Now, this very busy figure, which we're not going to go into too much detail, talks about the differentiation of our stem cells in the bone marrow that will divide off to become the mature erythrocytes, leukocytes, platelets, and so forth. And as they go through all these various steps, you'll, in another class of hematology, learn all these steps. Here, simply be aware that the bone marrow, when it's signaled, it makes cells that then specially differentiate into different lines. The 2 key words I want you to get out of this figure are myeloid and lymphoid. The white blood cells form these 2 pathways. The myeloid are the cells that are going to form and stay in the bone marrow and go straight to the bloodstream. Lymphoid are the cells that start in the bone marrow and then go to lymphatic tissues, spleen, thymus, lymph nodes. And so they will form the lymphocytes. Whereas you take a look at the granulocytes, and we take a look at the eosinophils, neutrophils, basophils, and mast cells, those are myeloid. Those 2 terms, myeloid and lymphoid, will come back to us later. Pay attention to those. All right, let's go specifically into the lifespan of each formed element. Once it's released into the blood, erythrocytes can last about 120 days. It's not really long, but bear in mind that erythrocytes have no nucleus. They don't have any DNA to be able to repair any cellular proteins or membrane proteins. They'll break down quickly. And so they're good for about 120 days. Now, lymphocytes and monocytes, when they go in the bloodstream, they can last for months to years. So the relative amount of lymphocytes in the blood is actually very small. Once they're activated, once they're signaled by other cells or by cell-to-cell hormones, now their lifespan shrinks tremendously because they become very active and they start burning up a lot of their cellular machinery. 7 to 14 days. Granulocytes, we're looking at our eosinophils, neutrophils, basophils, mast cells. Again, in the inactive state, they'll float around in the bloodstream for months to years, but once they're activated, very short lifespan. Platelets are little bags of chemicals that help us with blood clotting. They're even less self-sufficient than erythrocytes. They only last for a couple of days. So your bone marrow, those big megakaryocytes, are constantly pumping out platelets nonstop. All right, we're going to focus the rest of this video lecture on the erythrocytes themselves. A typical red blood cell is approximately 8 micrometers in diameter. The textbook actually mislabels this and says 8 meters. Oops. No, it's 8 micrometers. It's this red biconcave disc full of hemoglobin. Nucleus has been removed out. If some of the nucleus remains, when it's pushed out into the bloodstream, this is referred to as a reticulocyte. A reticulocyte is an immature red blood cell. It's full of hemoglobin. It's still functional in the regard of being able to carry oxygen, but it has a little bit of nuclear material in it. It by itself is not bad, but we always count the number of reticulocytes in our bloodstream because if we see an elevated reticulocyte count, that means something's weird going on in the bone marrow, and we're pushing these out of the bone marrow faster than normal. So reticulocyte itself is not bad, but we always look at that blood test, and if it goes high, it means look a little bit closer to figure out why. Problems with the erythrocytes really fall into two categories: too much or not enough. If your hematocrit is abnormally high, this is referred to as polycythemia. And I say abnormally high. If you live at high altitude like Flagstaff, Arizona, 7,000 feet in altitude, your body's going to naturally make more erythrocytes to carry more oxygen because of the thinner air. You would have a hematocrit higher than 45 normally. But if for an abnormal reason you have a high hematocrit, this is referred to as polycythemia. We'll get into more details of this in a second. On the other side of that is anemia. Anemia is not so much low red blood cells, it's an insufficient ability of blood to deliver oxygen. But for some reason, the blood's not functioning well. We'll look at examples. We see our typical red blood cells, these little biconcave discs. They soak up oxygen in the lungs. They go out to the tissues and they release it like sponges. They soak up carbon dioxide, go back to the lungs, release it, pick up oxygen, back and forth, back and forth. The molecule that makes up the inside is hemoglobin. Hemoglobin is made of these four big globin proteins, and this little saucer called heme, that heme pigment, oops, let me back up, that heme pigment has an iron atom in it, and this allows us to actually grab onto that oxygen and carry it through the bloodstream. Now, for polycythemia, this is an abnormally elevated hematocrit. Polycythemia vera is when there's some problem with the bone marrow, possibly a malignancy, a myeloid leukemia, something is overproducing red blood cells. This tells us that, yeah, there's something going on there. We'll have to do maybe a biopsy to try and figure out what's going on. Worried about bone cancer kind of stuff. Blood doping is an artificial polycythemia. We inject a person with a hormone called erythropoietin. This is a hormone you naturally make that tells bone marrow to make more red blood cells. If you inject yourself with more of this, then you get higher hematocrit, which if you're an endurance athlete, the highest hematocrit you can go is good, with the caveat that too thick of blood is hard to push through capillary beds and could cause strokes. And so that's why we're worried about people blood doping. It's very, very unhealthy. On the other side of that, we said was anemia, insufficient ability of blood to deliver oxygen. Hypoxia is a decreased oxygen level in the tissues. Now, that's a result of the anemia. Make sure that you understand the difference between the term anemia and the term hypoxia. I get students that want to mix them up all the time. Anemia is the insufficient ability to deliver. Hypoxia is the result. So watch the wording on my test questions so that you give the correct answer. Lots of things can cause different types of anemias. I'm going to give you a long list of them right here. All these different types of anemias are due to some different foul up in the system. Your take-home assignment, I want you to read through each one of these, find the cause. What is the cause that results in this kind of anemia? And so read through these, make sure that you can describe what is wrong in this person to result in this kind of anemia. An example is iron deficient anemia. And that simply means that we don't have enough iron to carry the oxygen. The blood does look kind of like orange juice. This color is correct for this condition. This blood is just not as good at carrying oxygen because it's low in iron. Sickle cell anemia is a hereditary situation where a person has a mutation of a gene that causes the deposit of an amino acid. The whole globin protein bends, and instead of that nice biconcave disc, we see this weird little sickle. And these guys will stack on each other and they'll plug up capillaries, and that'll prevent oxygenation downstream. There's another example of normal blood on the left side and sickle cell blood on the right side. That's the quick and dirty of erythrocyte problems, too much or too little. Take your time reading through that. Make sure you write out good notes, be able to describe the different types of polycythemias and the different types of anemias. Let me know if you have any questions. Good luck.